Hello guys and welcome back to Accenture Academy. So today we will be discussing further about in the microbial physiology. So in the previous lecture we discussed like how like what are the different phases of the growth curve, lag phase, log phase, stationary phase and the death phase, right? Now little bit mathematics of the growth. So this also guys you know already where generation time can be calculated by using this formula, right? So number of division is 0. It means there is only one bacteria is there. If 1 it is there, then 2 raised to 1, 2 you will get. So this is how it is going exponentially, right? Nothing new in this. So the time required for the population to double in size is the generation time. It varies depending on the species and also the environmental condition, ranges from 10 minutes for some bacteria to several days for the eukaryotic organism. So we know that like at least in the case of the eukaryotes, a mammalian cell, 24 hours is required in order to double. So this is how the you can actually plot like the minutes of incubation and the number of cells. Okay, no questions from here. Only the questions can come is like what is the generation time if initially these many were the bacteria and after these many hours these are the total number of the bacteria. So what is the generation time in between? So those that n thing you have to calculate. Then simple thing, you have to just equate this to the number given and then calculate with respect to that what is the n over there. Okay. Yeah, so this is how we are getting it actually. So the measurement of growth rate and the generation time. So let's say that N0, let's say that the N0 is the initial population. T is, NT is the population at time T. Okay, where the small n is the number of generation in time T. Now, the population which is reproducing by binary fission will be given like this. So NT population at time T, let's say that after 1 hour. So initial population into 2 raised to n. This is the formula which you are going to use. Now when you solve it, like they have applied the log, you will get log NT is equal to log N0 plus n log 2. Log 2 की value क्या है? 0.301, right? Now small n is equal to log t minus ये यहाँ चला जाएगा log N0 divided by log 2. So you will get this value. Now the mean growth rate, which is represented by mu, it is the number of generations per unit time. So mu is equal to n by t. So this value again divided by t. ठीक है. So this is how you will get the mean growth rate. यहाँ पे आपको क्या मिल रहा है? Generation time. Growth rate. Growth rate क्या होगा? Generation time divided by the number of generations per unit time. Okay, n by t. I hope you are getting it. So हमने क्या सीखा? n. Number of generations कैसे मिलेगा आपको? Log nt minus log n0 divided by आपका log 2. ठीक है. ग्रोथ रेट कैसे मिलेगा? n by t. याद रखना. Now calculation of the mean generation of the doubling time. So if a population doubles, then nt is equal to 2 raised to n0, यानी कि exactly double हो जाता है. Then you can use this formula. So where mu is equal to n by t we are using, and here you are getting this value. ये अगर नहीं भी पढ़ा तो चलेगा, but these two are important. Okay, are you able to follow, Krishita?
Yes, sir. Yes, sir.
Only these two are enough to understand like for the in future calculation. This is just a figure like how a cell is dividing. So you know about this. A young cell is there at the early phase. It starts getting all the nutrition required. Okay, so once the nutrition is available and the correct growth factors and environmental factor is in favor, then the cell will start preparing for the division by enlarging its cell wall, increasing its volume or the proteins which is required for the synthesis. Okay, then the septum begins to grow inward, chromosomes will move towards the opposite pole. So if you remember, the bacterial chromosome will double. Okay, like normal division will take place, and then please do remember that when after division they are actually like this, right? Concatemer they are forming. What is happening then? Topoisomerases are coming and removing these concatemers so that they get separated like this. Okay, and then they will start moving towards the opposite ends of the cell. Okay, now so then the septum is formed, and then slowly the contractile ring will be formed, and then it will help in the divisions. Okay, so they will go in the separate. Similarly for your plasmid also, like even plasmid also get replicated and then they are divided so that they can go in the opposite directions. Okay, now what additional thing you have to remember here is that the chromosomes or the plasmid do not move by itself. Now, who can tell me how it is then possible moving towards the opposite sides of the cell? Chandani, any idea? Hmm? See, it won't go by itself, the chromosome or the plasmid, right? It has to be done by someone. वो कौन है? Krishita?
Is it cytoplasm?
Motor proteins, something like that.
Yeah, so they're analog. Okay, so motor proteins, they are present in the eukaryotic cell. Same way, the bacteria also, they have got certain different proteins. Okay, and other categories is chaperones only. So which will make ensure which will ensure that half things goes in this direction and half thing goes in the other direction. Okay, so again, it is helped with the motor proteins only, but in the case of the bacteria, they are the analogs. Good. Now, prolonged decline in growth. Now over here, the bacterial population will continuously evolve. So what does it mean? It says that over period of time, okay, over period of time, one particular population is there. It will slowly grow. It will reach its maximum and it will decline. Again, out of that particular population, few bacteria will again emerge, will go and decline. So what is happening over here? From the same population, certain bacteria, okay, the group of bacteria, they are emerging, okay, they are evolving every time. So what is happening? Though overall population size, as you can see, it's almost maintained same, okay, but actually क्या हो रहा है? As there is generations taking place, okay, new generations are coming. What is happening? इसी में से बचा हुआ again it is coming and rising, again it is coming and rising. Okay, so what is that favor? It is the natural selection. See, when they are in certain environmental condition, it's not that the environmental condition is going to be same or constant throughout, right? As your environmental conditions are changing, let's say the introduction of antibiotics are coming or any toxins are coming because of some, human interference or whatever, what is happening? From a group of population of the bacteria, rest all are dying, but few are able to survive. So now those survived ones will increase in population and over period of time, they will decline. Okay, but again, certain group of bacteria, they will emerge as the survivors. For the new environmental conditions, they will grow and then again decline. So ये क्या है? ये आपका cycle चलता रहेगा. ठीक है? And that's how, we now have got the MDR strains. This is also known as arms race. Arms race मतलब क्या हुआ? If I am improving the quality of a drug to kill the broad size spectrum of bacteria, then I am at higher position कि मैंने ऐसा drug बनाया है that is killing all the bacteria. But at the same time, among the bacteria, there are certain genetic mutations taking place, which is helping few of them to survive this drug action. Correct? So what is happening now? Even they have evolved. अब क्या मेरा drug इनको नहीं मार पा रहा है? They have evolved. Again, those infections are coming. What is the next step then? I'm going to make a drug in such a way that it is going to kill this particular bacteria. So abhi मैं आ गया हूँ. Now what will happen? There will be few other mutations happening so that the bacteria is able to survive the action of this particular drug. तो ये आगे जा रहा है. then again like this, and then again the bacteria will go ahead. So this is the arms race, and ये कभी खत्म नहीं होगा unless until one of the species is getting extinct. Clear? This is clear, guys, to everyone. This will eventually come up when we'll do the ecology evolution part. Yes, clear? Chandni?
Yes, ma'am.
Okay, it's important to understand also. See, based on this only the interview questions might come. Okay, जैसे कि हम पूछते हैं, what if, you know, let's say एक example देती हूँ which we have asked, you know, once during one of our sessions. So there, let's say that there is a bacteria A population and B population is there. Okay, now there is a drug. A and B populations are like almost similar. Okay, let's say Bacillus species 1 and this is some other species 2, but Bacillus only. Now there is a drug, let's say drug C is there. It is able to kill your Bacillus species 1, but it is not able to kill 2. Okay. Now what can you propose as a scientist in order to use the same drug and to kill species 2 as well? Okay. Yes, Akshaya, would you like to try first?
Ma'am, can you repeat the question?
Yes, I'll repeat. So here what I'm asking is, there are two species of Bacillus, A and B. Okay, two different populations are there. Species 1, species 2. Like this is some species of Bacillus. This is another species of Bacillus. Okay. Now there is a drug in the market which is able to kill A. Okay, but not able to kill B. Now, as a scientist, what can you propose in order to make drug C able to kill B as well? आप ऐसा क्या करोगे जिससे same drug I can use to kill B also? As a scientist, what are your thoughts? Shruti?
Ma'am?
Yes, any thoughts?
Ma'am, the part of the gene that has developed drug resistance that needs to be checked and that can be removed. Like we can compare the genome, right?
Yes. So this is one step. Yes, you can do that, but not removed actually. What you can do over here is, first of all, that word, the comparison is correct. Like you are going to compare between A and B, like why the drug C is able to kill A and not B. Okay. First of all, that particular thing you're going to analyze. Let's say that the binding site of this drug C to A is something like this. Okay. Binding pocket. So the drug is able to bind over here and able to kill A. So you're going to compare. You can also compare the genome and the proteins, because eventually it is the drug which is going to bind to certain proteins only and inhibit. It can be any protein or enzymes by which the drug is binding and inhibiting the growth of this particular bacteria. Okay. So of course, yes, you can compare first between A and B. Compare like why it is able to kill A and why it is not able to kill B. Okay. Second thing, what you can do is something known as the SAR. Have you heard of SAR? Structural activity relationship. So in this, what to do is that you understand like how the structures are interacting. ठीक है? Let's say that A में आपके पास दो domain थे, okay, 1 and 2. But in the case of B, additional domain, something like this is also required, because of which drug C is not completely able to bind to this particular thing because this domain is missing and hence not able to kill B bacteria. Okay, what you can do as a scientist is that you can add this particular domain in the drug. तो मुझे अभी पूरा का पूरा ड्रग सिंथेसाइज नहीं करना है. What I have to do? I have to just add another functional group. Okay, so that it can have over here also binding. And now it is able to kill B also. So मुझे उतनी मेहनत नहीं करनी पड़ेगी. Like from the scratch, I have to search for a new drug which will kill the bacteria. Okay, screen करना पड़ेगा कि कौन सा drug करेगा. And then I have to understand the mechanism of action. Like कहाँ पे कर रहा है. It's always better that I go with this R, structure activity relationship. I'll understand, I'll compare, I'll understand कि ऐसा क्या है जो drug C is able to kill A but not B. I'm going to compare it, I'm going to understand it, and once I understand, okay, this is the additional functional group because of which it is not able to bind, or it can be the other way around कि एक ही domain की जरूरत है, इसकी जरूरत नहीं है. So I can do the modifications on your drug, either add functional group or I can delete functional group. Okay, from the drug C. Later on, this drug C is also able to kill your bacteria B. Okay, now when I say add and delete, of course, it's all your organic reactions. Okay, this is all your organic chemistry. Clear?
Yes, ma'am.
Okay. Okay, so the generation time, so they have calculated over here. Now, how do you calculate generation time? ये तो नहीं कोई पूछेगा, but still we should know. So when you plot a graph between the time and the number of cells, okay, so here number of cell is given like 1 into 10 raised to 7 में दिया हुआ है. So now when you plot a graph over here, as you can see, lag phase है, उसके बाद log phase है, and eventually it will come like this. ठीक है? अब मुझे generation time calculate करना है, or the doubling time. So what I'm going to do is I'm going to see, look at the population where exactly it is going to become double. So 0.5 से 1 double हुआ है या 1 से 2 में लूंगी. ठीक है? So I'll plot like this. I'll get the lines like this. I'm going to get the time. Okay? So this time I'm going to get is the generation time. Clear? Simple है, graph से आपको मिल जाएगा, no issues. So this is example where each generation time is given. So incubation time of bacteria, so 40, bacillus 40, S aureus 37 degrees Celsius. This is E. coli 40 degrees Celsius. Now generation time in hours it is given like 0.35, 0.43, right? So like in many cases we say that the bacteria, E. coli especially, it takes hardly 20 minutes. But it is not exact 20 minutes because as if you remember when we were talking about the DNA replication, E. coli में क्या होता है? The moment one replication is happening, okay? What happens? तब तक आपका second replication शुरू हो जाता है. Okay, so that's why it is taking very less time. So before one round of replication is getting over, the second replication round starts. So that's why in the case of E. coli, it is fast, the doubling time. Okay, ये आपको याद करने की जरूरत नहीं है. That's only for the understanding. Okay, now how are you going to measure the microbial growth? So I got this slide. It's really very interesting and very helpful also. So guys, आपने lab में कैसे किया है? How have you started with the microbial growth? Like what all methods do you know apart from these measurement of cells? These are the direct methods. What are the indirect methods of counting? Anyone?
Ma'am,
Huh.
so we did it in the lab a few days ago. So we took the stock bacteria which had an unknown number of cells.
Huh.
We plated them first and then created a match. And then we collected those back and then
Hello. Hello, Shruti. Huh.
Ma'am, can you hear me?
Yes, yes. Please
Yeah.
repeat.
So
Huh.
and then we used N1 V1 is like equals N2 V2 to figure out how many colony forming units are there.
Yes, correct. So that is the indirect way where actually you get the colony forming unit. Okay, good. So this is your first lab experience?
Yeah.
Okay, good. So finally learning. Yeah, good. So that is the indirect way where you actually go with the colony forming units. Let's say that you don't know kahan pe kaun sa bacteria hai, kitna hai, no idea. Like, or most of the time we take the soil sample also, right? इस particular soil में कितने bacteria है, that you can do by colony forming unit method से. In that way you can calculate whether it's a soil or any sample you take. But do remember that you have taken the weight of the soil, like 1 gram से चालू किया है या 10 gram से चालू किया है, because then we can say 10 gram में अगर इतने बैक्टीरिया थे, so 100 gram में कितने होंगे, so you can just back calculate. Okay. So measurement of the microbes, it can measure changes in the number of cells or mass of the population. Okay. So if it is the cell numbers, we have got the direct cell counts, counting chambers are there, electronic counters are there, and using the membrane filters. Counting chambers, I'm sure you must have done. Hemocytometer में भी करते हैं, if you know how to read this thing. Any idea how to read this chamber, counting chambers? किसी को आता है?
Yes, ma'am.
Yes, Shruti, can you explain?
Yeah. So we try to spread the cells as well as possible, usually by using a diluent, and then we count 5 of the like we count 5, what do you call it, areas within and take an average of it.
Okay, so in this hemocytometer, like what all quadrants do you consider and how do you calculate the number?
They are edges, on the edges, the 4 corners.
So this one?
Yeah.
And the middle one. Okay. So
yeah.
how do you do is that you actually start counting this. Okay. Let's say you are adding this. 1 second. Let me erase it. Okay. So basically, let's say I'm gonna calculate on this. So 1, 2, 3, 4, 5, 6, 7, 8, 9. ठीक है. कभी कभी क्या होता है, the bacteria is present over here. ठीक है. Then confusion comes कि इसको हम इसमें ऐड करें या इसमें ऐड करें. So whatever it is, add in your first case. So then don't add in the second. Are you getting it? अगर वो लाइन पे है, किसी एक में आपको उसको ऐड करना है, so that you don't take it in the other way around. ठीक है. So then you calculate this. ये chamber, ये और ये. You keep it over there, and then we take this also, the 5th one, where this is in like 25 small squares are there. So here you're going to calculate again. Now whatever you get, you add it, and whatever the dilution you have taken, that also you have to consider. ऐसा नहीं है कि उस particular sample में सिर्फ इतने ही बैक्टीरिया होंगे. Let's say that there are 105 are there, but then how many times have you diluted? Okay, we load around 10 microliter only. So 10 microliter में अगर आपके पास 105 bacteria है, so how many times you have diluted, that particular value will come here. Okay, so those many cells are present in that much amount of your sample. Got it? Guys, एक बार try करना. अगर नहीं आता है, let me know. We can discuss it again with detailed example. Okay, this is important because जब हम part C questions करेंगे, so these questions might come, the practical based questions. Okay, then we have got the electronic counters. ये क्या होता है? You have got the petri dish, right? You just put it into the machine and it will just counter. Then membrane filters, I'll explain as this as we proceed. So the counting chambers, this is very easy, inexpensive and quick. It is expensive if you break the hemocytometer again and again. Otherwise, it's very cheap. Useful for counting both eukaryotes and prokaryotes. Cannot distinguish between living and the dead cells. Yes, this is the disadvantage of the counting chambers. Even if the bacteria is dead, we cannot say that it is dead or not, because हमने एक बार उसको बाहर ले लिया and it is present over here. Then under the microscope, you won't understand whether the bacteria is alive or not. Okay. Now counting on the membrane filters. So cells which are filtered through the special membrane that provides dark background for observing the cells. So इसमें क्या रहता है? You have got, I don't know whether you have seen the filtration flask or not, something like this. ऐसे कुछ रहता है. Okay, तो हम यहाँ पे membrane लगाते हैं. यहाँ पे suction रहता है. So then you put your sample over here. Membrane is present. So now I'm gonna do the suction. So suction की वजह से क्या होगा? Through the membrane, the liquid part of the water or the media will come here. ठीक है, वो नीचे आ जाएगा. And membrane में आपके bacteria stuck हो जाएंगे. ऐसे. ठीक है. This is happening in the vacuum. Okay. Now then you can actually calculate like how many bacteria are present on the membrane. So cells are stained with the fluorescent dyes, and then you can actually calculate. There are certain dyes which can distinguish between living and the dead cell. So you know there are certain dyes which will be taken up by the living cell. So जो आपको stained bacteria दिख रहे हैं, those are your living cells. Rest all it will be your dead cells. Then we have got the FACS, flow cytometry, or also known as the FACS, fluorescence activated cell sorter. So here you can have the microbial suspension, which is ऐसे रहता है एकदम, it's a very small say small and narrow nozzle is there, so which ensures that at a time 1 or 2 bacteria are dropping like this. Okay, then based on the laser, if it is detecting, then it will say okay, this is a bacteria. So movement of microbe through that orifice impacts electric current that flows through the orifice. Okay, so it will tell okay, 1 bacteria, 2 bacteria, and it is very fast. Okay, it's not like ek ek karke rehta, it's very fast. So whenever there's a disruption of the current, the flow, whenever bacteria is coming, that is taken as 1 or 2. Okay, then we also have the antibodies which can be used to determine the size and the internal complexity of a particular cell. So these are the methods by which you can directly count the bacterial cells. Okay, this is also applicable for your eukaryotic cells as well. Now, as I said that in the direct counting methods, the disadvantage is that we cannot differentiate between the viable and the dead cells, right? So now we can have the other methods like spread and pour plate techniques. The thing is that it just takes more time. Okay, इसमें बहुत time लगता है, but then if you are in the microbiology lab, then this is like routine work and it is important also in order to start any of your experiment. Okay. So counting method. So we have got the spread and the pour plate techniques. So whatever sample is given to you, जैसे कि कभी-कभी क्या होता है, आपको freeze sample दिया है, या you are bringing from a very extreme conditions like very hot area से आप लेकर आए हो, and then or maybe cold area से, whatever the region, or you can have a soil sample also. Like usually we start with this only. So when I was doing my MSc project at Delhi, so that person, he asked me, my scientist, okay, before coming, he said, why don't you bring a deep sea water ka sand, not sand, what to say, the soil from the deep sea water. Okay, and I was in Kerala, so I was like wondering how. I mean, of course, then my dad was in Navy, so it was easy. So he asked one of his junior, the diver. Okay, so he went very deep and he got the sample for me. That sample I took to Delhi. Okay, you won't believe, even I was not having the good idea like kitna sample lagega for the bacterial isolation, because we wanted a unique bacteria which was not mentioned or discovered before, something like that. So of course when it was deep sea water, so that condition was halophile. I hope everyone knows about it. What are halophiles? Yeah, so I have taken the sample, and from Kerala to Delhi I carried around 5 kg of soil. Okay, and now in the lab you don't need that much of soil. I say 1 gram was also sufficient just to isolate the bacteria. But since even I didn't know like kaise shuru karte hain experiment, so I carried 5 kg of soil, which was stinking so bad. that sea ka smell and all. So anyway, so that was my experience. When I went to the lab, I carried this 5 kg of the soil in the lab, and he was like, "What is it?" I'm like, "Sir, you asked me to bring the soil." Yeah, but not 5 kg, 5 gram would have been more than enough. But yeah, it's all about the experience. Like, even I don't know, I didn't know like how to start the experiment. That was my first time. So then, of course, I took 1 gram of sample, I weighed 1 gram of sample, then I diluted it. So I diluted with, of course, your deionized water. What is deionized water? Free of ions, free of bacteria. Okay, there's nothing else is present. So then you remember how much dilution you're doing. Let's say that I made it into 100 ml. Okay, then from 100 ml, I'll go for the serial dilutions. I hope everyone knows what is serial dilution, right? You 10 raised to 1, why 10 raised to 2. So from this 100 ml, I have taken to 1 ml here. From here, I mix it again, taken 1 ml to and added to this. Then again here, like this. So the dilution factors are like in the negative then. Okay. So I dilute the sample of the bacteria, then it is spread over the solid agar surfaces or mixed with agar and poured into the petri dishes. So these are the two ways. One way is where you can prepare your nutrient agar plates. Okay. So what I'm doing, मैं यहाँ से अभी sample लूँगी. Guys, this much is clear, like what I have done here, experiment wise. Is this much part is clear? Practically?
Yes, ma'am.
Okay, cool. Now, what I'm doing, so I have got around 10 raised to 1 से मैंने 10 raised to 8 तक, okay, dilution. I have 8 test tubes, okay. So यहाँ से 1 ml लेके यहाँ डाला, यहाँ से यहाँ डाला, and till there, till 10 I have done, till -8 I have done the dilutions, okay. Please remember 1 gram of the sample, 100 ml already add किया था. Then from here I have started doing the dilutions again. So I'll take around 1 ml or 0.5 ml and I'll spread onto the nutrient agar plates. So spread plate kaise kiya jata hai, right? So I'm gonna do that. Or okay, this is your spread plate method. Or what you can do is you have got the what to say agar is there. Okay, तो जैसे वो थोड़ा सा normal temperature में आता है, like which can survived by the bacteria, you add into that and then pour onto the plate. So what will happen? Spread plate में आपको on the top you will see if any bacteria is growing, right? On the top. Pour plate में you will see on the top as well and also in the inside of the agar. So both methods are there. Same way I'm going to do it. Okay. Now after incubation, the numbers of organisms are determined by the counting the number of colonies and finally multiplied by the dilution factor. Okay. So अगर मुझे suppose इसमें से मिला, if suppose this is like the colonies are overlapping with each other, you're not able to distinctly count the colonies, then I'm not going to consider this. क्या होगा? Around 10 dilution of -5 and -6, let's say -8 itself, I'm able to count very nicely. Okay, there are 4 colonies are there. ठीक है? So they are distinct. So I'll say that in the 1 gram of the sample, I have got 4 into 10 raised to -8 dilution factor. Okay, not 8, sorry, +8. So I'm going to have these many colony forming units then. Get it? Okay, so that's how you're going to calculate. Now membrane filter technique, ये एक technique हो गया. So colony उसी में ही बनेगा when the cells are alive, right? So this is the viable counting method, both spread and the pour plate method. Now coming to the membrane filter technique. So if you take the bacteria from the aquatic samples, they are trapped on the membranes. Okay. Now you put this membrane onto the culture media. ये membrane में आपके bacteria लगे हुए हैं ऐसे. They take this membrane and place it on the top of your nutrient agar. Remove the membrane, allow it to incubate. Next day, you will see that beautiful, so many colonies are there of so many different colors. Why different colors? Each bacteria has got its own color, right? It's from the aquatic sample. So सारे बैक्टीरिया होंगे, कुछ actinomycetes भी होंगे, or it can be anything. Fungus भी हो सकता है, you never know. So colony count determines the number of bacteria in the original sample. So you get all this thing. So it is very difficult to count. So what you're going to do, you're going to dilute it again. if it is not overlapping with each other. Okay. Yeah. So here see you the apparatus is shown. So here आप एक membrane लगाते हो, ठीक है, यहाँ पे membrane रहता है, and then it will pass through the membrane. Suction एक लगाते हैं हम यहाँ पे, it will have suction over here. So because of which what will happen, the water will pass through the membrane, and जहाँ पे भी bacteria रहेंगे, it will get stuck onto the membrane. So now this membrane you take it and place it onto the nutrient agar petri dish. Another day you will see that typical colonies are present. Same here. You have placed it on the media, and next day you see the colonies. So if microbe cannot be cultured on plate media, the dilutions are made and added to the suitable media. So this is also important, guys. See, कभी-कभी क्या होता है, कुछ बैक्टीरिया ऐसे होते हैं जो आपके न्यूट्रिएंट मीडिया में, सॉलिड मीडिया में ग्रो नहीं करेंगे. Then you have to go with the nutrient media support. Okay. So then there it is the turbidity which is going to determine the most probable number. So then their turbidity कितना ज़्यादा turbid होगा, then you can actually calculate if this much is the OD I'm getting. So there also the one like each lab has got their own record. If this is the 0.1 OD at 600 itna hai, so these many bacteria are there. If you have got the OD 0.8, then these many bacteria are there. Okay, so that's how the calculation is there. So determining based on the turbidity. Now coming to the measurement of cell mass. Okay, dry weight. It's a time consuming and not very sensitive. There is no point of taking the dry weight of any bacteria. Then quantity of a particular cell constitute, like kitna protein, kitna DNA hai usme. This becomes important if you are going to do experiments based on the DNA and protein base. Okay. Now turbidometric measure, this is based on the light scattering. So it is quick, easy and sensitive as I mentioned. So you are going to check the OD at 600. Jitna zyada light scatter hoga, ठीक है, उतना ज्यादा आपको पता चलेगा कि हाँ turbidity है. Like I can show you here the light scattering or the absorbance. See, when the cell is getting more scattered and the absorbance is less, what does it mean? Less bacteria, right? More the cells are there, so more light will get scattered. So more light getting scattered means utna zyada turbid hai aapka sample, and hence number of cells are going to be more. So 0.21 absorbance means that there is more light as compared to the absorbance of 0.04. This everyone knows. I'm sure you might have done in the lab also. Is it clear, guys? Everyone? Chandani?
Can you repeat the question?
So question was, have you done in the lab, like, taking the OD of a particular bacterial sample? So where you can understand more the absorbance means more bacteria is present because the sample is going to be very turbid.
No, ma'am, I don't have lab experience.
Okay, no issues. So just understand that when you have got the absorbance more, it means more bacteria is present in the sample. Okay. Now, then we have got the continuous culture of microorganism. Now, what is a continuous culture? It means we keep on adding the new nutrition. Okay, new batch of your nutrition is there, and always we remove the waste. So because of which the cells are always in the log phase, and we get a continuous culture system. ये हम कहाँ करते हैं? Like in the industries and all, where we always want, the cells growing. For example, I hope everyone knows about the Biocon company, which is there in the Bangalore. अगर नहीं पता है, so that's okay. I mean, there are many companies. Why I'm saying about the Biocon is because it was started by a lady with 100 rupees in her hand. So it's a very great story about her, Majumdar Shah. So she started this company and she started by producing insulin. ऐसा नहीं था that she was doing something great or something new. It was already there in the market. Okay, insulin was there in the market. But what she did was like making sure that the microbiology concept what she had, okay, she was, I think, MSc microbiology, something like that. So what she did was like So the insulin, all genetic engineering was done and people were using it. Was just very, very costly. So what she has done is making sure that the bacteria is always in continuous production of the insulin. Okay, and making sure that once the bacteria has been taken, the downstream processing. Okay, so what she has done is genetically modified in such a way that the bacteria is synthesizing insulin and secreting out. So यहाँ पे क्या होगा? The bacteria can be reused again, right? So what I need is the media then, extracellular media. So after some time, when I say that removal of waste, so we are removing the used media. मीडिया बाहर निकाल रही हूँ, उसमें हमारा इंसुलिन भी प्रेजेंट है. Okay, so now what I'm gonna do? I'm gonna go downstream processing where the protein purification step will be taking place. So then we have got so many steps, और ये सारे काम हो रहे हैं आपके tons में, not in liters. It is happening in tons. So you can understand like how much bacteria and how much, you know, nutrient media is being prepared, autoclaved, sterilized, and you know, it is put into the container. Big containers are there, 1,000 liters, or I don't know. I have not seen actually, but I have just heard only. And then at the end, by the time it is reaching the more steps and all, it goes into the HPLC. जहाँ पे सिर्फ एक single peak आएगा, which is corresponding to the insulin. Now then that is packaged in 1 ml का syringe जैसा जो रहता है, and then it is sold into the market in a cheaper way. That's why now even the below poverty line people, if they have got the diabetes, even they are able to afford. Okay, so it's all about the knowledge of microbiology. Okay. Importance of continuous culture method. So the cells are always in the log phase. So there is a constant supply of the cells or whatever drug you wanted. Like it can be antibiotics, not only insulin, it can be any antibiotics. So study of microbial growth at very low nutrient concentration, close to those present in the natural environment. Study of interactions of microbes under the conditions, which is resembling to the aquatic environments, and food and industrial microbiology. So, you know, just now I have explained this thing. Now the chemostat. So chemostat means it is the rate of incoming media is equal to the rate of removal of medium from the vessel. So जैसे ही आप जितना nutrition डाल रहे हो उतना ही आप उसका waste भी निकाल रहे हो. So the total overall volume is going to remain the same. Okay. Now chemostat operates best at the low dilution rate. So what does it mean? It means अगर मैं volume जो add कर रही हूँ, it should be less. इन अगर 1 टन का आपका चैम्बर है, सो उसमें इफ आई एड अराउंड 10 liter of new media and 10 liter of waste I'm removing, then it's fine. ऐसा नहीं होना चाहिए कि मेरे पास 1,000 liter of 1,000 liter of culture was going on, right? And in that I added new 800 and I removed old 800. ठीक है, ऐसा नहीं होना चाहिए. Then it's a shock for the bacteria. Instead of this 80 I can add. Okay, did you get it now? The dilution rate over here.
Ma'am, can you explain this again?
Yeah, so see, let's say that I'm doing the culture of 1,000 liter. Okay, what I'm saying is that instead of adding new 800, this is only media, right? And यहाँ से मैं old 800 liter निकाल दे रही हूँ. So with the help of those 200 liter bacteria, it can survive on the 800 new media क्योंकि उनके पास कितना nutrition आ गया है, right? They can survive. But when you do such dilution, it's a shock to the bacteria कि हमारे like it's the same thing ना, all of a sudden the people are missing. क्या हुआ? Even though resources are there, why then the people are missing? Same way, the bacteria will also understand so much of nutrition is available, then why the population of the bacteria is less. So the rest of the bacteria, they get in a shock. So they get, this, they're in the stress now. कि कहाँ गए हमारे बाकी के बचे हुए bacteria when the nutrition is available so much. So it will take time for them to readjust, to understand, and then again regrow. Okay. But if I do 1,000 liter में, if I add new 80 liter of fresh media and remove 80 liter, then people won't people, I'm saying the bacteria won't recognize this loss of 80 and the newcomers 80 in 1,000 liter. Now did you get it?
Yes, yes, sir.
Okay. So not the bacteria, but when you take it as an analogy with the humans, कि 8 नए आए हैं और 8 पुराने गए, इतना फर्क नहीं पड़ेगा. बट पॉपुलेशन अगर 10 की है, इसमें अगर 8 चले गए तो आपको फर्क पड़ेगा. वो जो 2 पुराने लोग हैं वो यही सोचेंगे ना कि हम अकेले पड़ गए हैं, हमारे साथ कुछ भी हो सकता है. These 8 people are new people, they can do anything, right? So it's a shock for the existing ones. So that's why it should be done at the low dilution rate. Okay. So nutrient concentration, cell density or biomass, it will decline. Okay. When you all of a sudden increase the concentration, new concentration आप यहाँ पे ज्यादा बढ़ा दोगे तो क्या होगा? Cell density आपका वहाँ पे कम हो जाएगा. Generation time भी आपका कम हो जाएगा. Now the turbidostat, it regulates the flow rate of media through vessel to maintain a predetermined turbidity or cell density. Dilution rate can vary, no limiting nutrient because you keep on adding the media over here. It operates best at high dilution rate. क्यों? Because see, it has to maintain its turbidity. जितना ज़्यादा turbid रहेगा उतना ज़्यादा वो काम करेगा. So यहाँ पे क्या है? You can keep on adding the media. Okay. And what happens is that when you add new media, what will happen? Already it is so turbid. They are waiting for new media to come so that they can start growing. Okay, so that is your chemostat and turbidostat. Okay, so here, okay, let us start. This is actually the new topic, the environmental factors on the growth of the bacteria. So guys, I hope everyone knows what is halophile, acidophile. We are not going to take this in, detail. Ki halophile means they can survive at high concentration of salt. Acidophile means they can survive at the extreme pH range, like of the acidic and the alkaline, that is alkalophile. Then psychrophile kya hota hai, mesophile, thermophile. I hope everyone knows all this thing. Yeah.
Yes, ma'am.
Yeah, so this is like something which you know, barophile, like which can grow at a high pressure. Then the examples are also shared, and I don't think anyone is going to ask, but it's always good to know, like what are the meanings of this at least. Okay, so psychrophile, which grows at the cold conditions. So I have worked on psychrophile and halophile, and working on psychrophile is difficult. It's because they don't grow itself. I mean, itna late grow hote hain, it takes around 10 to 15 days for them to grow. And obviously, they won't grow at the normal temperature of 37 degrees Celsius. Grow honge to cold temperature mein hi honge, but it will take so much of time. So working on them is very difficult. Okay. Halophile was also quite okay. You just have to add, point, 0.2 to around 1 molar of NaCl and the other salts, then it is easy. Okay. Yeah, so that's all guys for today.